But we thank Kim for coming up from Juniata College. Her collaborator down there, Juniata, sent her to us, so we're really excited to see what she has to talk to us about today. Thank you.
Right, so we're going to talk about seeing the forest for the trees, random forest, and fracking. All right, so who am I? Well, I'm a person who owns a Golden Doodle and has a rather large board game collection. So I'm a pure mathematician and an applied statistician. So my PhD is actually in mathematics. It is from Penn State. And so is the Master's in Applied Statistics, which I earned actually a few years ago, so post-PhD. I teach math, statistics, and data science, including data science at the master's level. And I do statistical work on various projects, including biological work. And as mentioned, I do work with Dr. Gina Lamandella quite a lot. But I also do a lot of scholarship and teaching and learning projects. I have currently one in effective homework feedback and calculus that's going on. That's not my data, but I'm analyzing it in the similar situation with our Common Read program. And I should state for the record that I'm not a biologist. In fact, you're my first biological audience. I've given this talk to various groups of undergraduates and their faculty, and they've been mostly mathematics and statistics students. So we'll see. I imagine you'll ask different questions. That'll be fun. All right, as I mentioned before, this is a project with Dr. Gina Lamandella and her lab. That's her on the left. Two of my students were involved with this project. Veronica Kirchner is a 17 grad of math and bio. The random forest works is hers, and Rebecca Drucker was math and CS. Veronica is currently a clinical research laboratory analyst in Wellspring and Lancaster, and Rebecca is a CS PhD student at SUNY Stony Brook. And of course, this is a grant-funded venture, so there is a lot of grants involved with this data. All right, so we'll start out, we'll look at the biological problem that we're going to examine. And then we'll talk about the statistical problem. And then what makes this an undergraduate talk is we're going to talk about where the statistical technique comes in. What is a random forest? How do you get one? How does it work? And then we'll get to a statistical answer and a biological answer. And then we'll talk about where this project is going now. So note that this is the general statistics pipeline. We'll mention that again later. But that's part of how this goes. All right, so the biological problem to start out with is, can we tell about samples that we get near and in waterways if there's active fracking going on nearby? So shale fracturing for natural gas extraction, if you're not familiar with it, this is how it works. You take a well, you drill down into the side, into the shale, you push in an amount of water, sand, and a certain number of undisclosed chemicals that are generally biofilms, biocides, excuse me, and it fractures the rock. That's the way it's called fracking. And then that allows the natural gas to be extracted. And why we talk about it here is Marcellus Shale, which is named after an outcrop in Marcellus, New York, generally has natural gas in it. This is the Marcellus Shale play in this area. So you can see the dark green is where there's actually thought to be natural gas. The Marcellus Shale extends out beyond that. The little yellow dots are all Marcellus Shale wells, are all fractured wells. Note that they're pretty common in PA and West Virginia, not in New York, because I believe they eventually outlawed fracking. But you can see there's quite a lot of region here where this is. Pennsylvania is the second highest producer of natural gas by fracking. The first is Texas. Uh-oh, that went the wrong way. Well, in classic PowerPoint, it is even heard as my thing. So if we talk about the streams, I'm not going to be able to fix that. PA should be this way. We're in, I'm in Huntington County, just south of here. Here's Penn State. The streams are up in Clarion County and in that environment. So let me look at a better map that hadn't flipped itself. So here are the streams. There were 31 streams in this project. 29 of them are in this picture. 2 of them fell outside the map. They were measured from 2012 to 2016. So this is time. There's some of these have been measured multiple times. The ones in green were never fracked. The ones in red were active fracking at the start of the project, and the ones in the purple were the ones where active fracking started while the project was going on. You'll notice they're named strange things. Pennsylvania likes to name strange things other than stream or river, so lick, hollow, fork, all those things, they're all streams. So this is our 31 streams that we measured. And then the data was collected on them, right? We have the sample, which stream is it from, what are we sampling from? Generally, we sampled from water, from dirt, the sediment, from bryophytes, so like mosses, and from biofilms, like foam. So not all of this was sampled from water, although a lot of it was, but it was all near waterways in each case. And so we collected things about active wells and well pads. We had pH and temperature for every sample. For the ones that were water, we had more chemical measurements. There we are. So that's our metadata. And we're going to look at the bacterial microbiome, right? So we're going to examine what bacteria are present. That isn't saying we couldn't have looked at the microbiome, the funguses, the virome, or other eukaryotic organisms, but we're just looking at bacteria first. All right, so we go through the general process, right? We take our sample, we extract the DNA. Since we're looking at bacteria, of course, we amplify, we make more copies of the 16S rRNA gene where it's differentially expressed by bacteria. So different bacteria have different segments. We can use that to identify which bacteria are there. We use Illumina for sequencing, run it through QIIME, cluster the things, and then we identify our organisms. This is older data. This is identified in operational taxonomic units, that's OTUs, because it doesn't quite work out to be species. You get subspecies and stuff in there. This is older data. We now use a slightly more modern pipeline and then identify an amplicon sequence variants instead. Yes, these. All right. So why are we doing genomic annotation? You probably already know this, but most mathematicians don't. There are thousands of types of bacteria in most samples, and we can't grow most of them on a plate. So we need to do this. And the diversity is big, so I don't expect you to be able to read this graph. But along the bottom here, this is one for each sample. And the colors represent the percentage of the phyla of bacteria that are in there. So even at the phyla level, this is a pretty diverse sample. So you can see that there's lots and lots of different bacteria. My personal favorite is the bottom one, which is labeled other, as in we know it's bacteria, but we haven't even classified it by phylum yet, which happens a lot in this land, or at least there's some unknown part. And that's the yellow along the bottom. So there's a certain percentage of unknown bacteria. All right, I mentioned we're following the statistical pipeline, right? This is based on the statistical consulting pipeline by Janice Durr. It's an oversimplification, I'll mention that in a second. But we started with the scientific problem. We're going to go now to the statistical problem, and then we'll make a statistical solution. Then we'll have to come back to biology. Right, and note this is an oversimplification of the interaction between statistics and laboratory science, because we're not talking about design or data collection or data management or any of that sort of stuff, which also lives in this land. But this is the part we're talking about. All right, so our statistical problem is we want to predict fracking, in particular active fracking tests, that the well is actually going, based on the microbiome and know which microbes are involved. Right, so our goal is to make a statistical model for prediction and description of the responsive fracking status. So this is what we call classification problem, right? Where what we want to predict is actually a category. Is there active fracking or is there not? And the technique we're going to use is random forests. And it uses what are called classification trees. All right, before we talk about random forests and classification trees, I want to mention for a second why we aren't doing some sort of regression. So if you don't work with bacterial data, you may not know this already. Right, we can make a model. So here's things that are actively fracked. Here's not. These are the reeds of Sphingobacteriales. You might want to make a, and yes, you have to practice saying this. You may want to make a regression here. Of course, we can't do regular regression. We have to do logistics or something. However, most regression assumes that your data is relatively normal, or at least the errors are, and the data is our substitute for that. Most bacteria, when you get them out of the data, look like this. They're zero heavy. They have outliers. They're really, really right skew. They are so not normal that we end up using non-parametric things all the time. That is the way things work here all the time. All right, so we're going to talk about classification trees and then use classification trees to talk about random forest. So first, I have to tell you what a tree is. Okay, so this is math, right? Tree is a type of graph. And we're not talking about the y equals x squared type of graph. We're talking about the dots and lines kind of graph. A graph is a collection of dots, they're called nodes, and edges, that's the lines. And a tree is a connected graph, that is, it's all one piece, where there's no way to go around in a circle. There's no cycles. So something that looks like this. So this is a basic tree. We got dots, we have lines, they collect them together. Note that it looks more like a tree if I flip it upside down, which is technically the same tree, but is traditionally drawn this way anyway. Note in this case, I've drawn it so each one has two edges off the node. That doesn't have to happen, but the algorithm we're going to talk about for making a classification tree works that way, so that's what I've drawn. And we're not allowed to have an edge like here, for example. If we had an edge here, we'd be able to go around in a circle. And there's a lot of vocabulary here. The bottom ones are called leaves. I'm likely to slip up and say that one. The top one's called a root. They are all bad jokes about it being named tree, which is kind of how mathematicians often work. So classification trees are a type of tree. They're a statistical thing. We're talking about statistical classification trees. So here's a classification tree for this thing, where we're trying to predict fracking status. So for example, if we have a sample with 5 Cytophageaceae and 12 Spingarobacterialis, we look at this tree and say, okay, is Cytophageaceae bigger than 10? No, it's not. Is Spingarobacterialis bigger than 6.5? Yes, it is. So our tree is going to predict that we're not fracking here. So this is a classification tree. This is a statistical classification tree. Note that the idea of classification trees is sort of a flow chart you follow. It's something that works like every time you pull a nurse line to ask them if you should be going to the doctor, they have a tree they're going through, whether it's written that way or not. All right. So in order to talk about how these exist and how they work, I'm going to do a simplified problem that's simpler than the thousands of bacteria we got. And so we're going to stick with, is it going to rain today? Should I have my umbrella with me? All right, so our response is going to be whether or not it's raining. And we're going to talk about possible predictors. Talk to your neighbor for a second. What possible predictors would you put in this? You're making this thing up? What do you do? Cloud? Yeah.
Yes. Ask Siri.
Ask Siri. All right. So what sort of things do we got? I heard a lot of good things out there. What do we got?
Is it gray out?
Is it gray out? Is it cloudy?
Temperature.
Temperature.
Weather app says it will rain.
The weather app says it's rain. Not a bad one.
Knee pain.
Knee pain. Ah, yes. Yes, that joint. Yeah, and I picked chance of rain and whether it was cloudy or not, because I could get those off of Weather Underground. I tried to make real data, and when I did this in the fall, I found that it was too rainy, and it rained a lot, and I couldn't make a good model from my data because it was almost all rainy, so I had to kind of fake the data. But right, I have data that looks like this. So I started from real data, and then I added some more not rainy days so that it would actually work. Notice, by the way, the chance of rain always goes by tens on Weather App for some reason. We like nice round numbers. All right, so let's talk about how we make a tree for this data to predict whether it's going to rain or not. So we start from a node. And at that node, I need to pick what variable am I going to split on, right? When we have these things, we split in 2 directions based on a variable. And we're going to pick the variable that will give us much less error. And we'll talk about what that means later. Right, and in this case, I have 2 choices. Am I going to look at cloudy or chance of rain? All right, so I picked it was chance of rain. We'll talk about how soon. And then I'm going to add a split to the tree for that variable. I'm going to pick a number, or for a category, categories, that go to one side or the other. So I'm going to choose what chance of rain goes which way or not. Where I want one side to be more likely to rain and one side be more likely to not. And then like most algorithms, we then lather, rinse, and repeat, right? We now pick a new node and then we repeat until we can't either make the error much less or we hit whatever depth of tree we want to go, however far down we want to go. The error much less is the most right, so in order to talk about how this works, we're going to need to talk about how we make splits, because that's how we determine about error. So first of all, let me talk to you about this graph. First of all, the y-axis here is a placeholder so that I can jitter the points so they don't all overlap each other. On the bottom, we got chance of rain. The red dots are where it's rained. The blue dots were where it didn't. Maybe I should reverse this. And the numbers here are how many dots lived in that land. Because chance of rain goes up by tens, you get more overlap than you would if it were actually really a continuous number. And the question is, if I'm going to draw a line here somewhere for chance of rain, where do I want it? Where would be the best split where I'm going to predict one side to be raining and one side to be not? And think about it that way. So talk to your neighbor, where do you make a split? Where do you want to make a split?
So we think it depends on how much you hate carrying an umbrella.
That would be true. Right, of course, choice of algorithm depends on your goal. So now that you've thought about it, here's where I started. I started with the it's always right when it says rain model. This is a great model for predicting rain. When it says it's going to rain, it's going to rain, at least for the data we have. So it's wrong 0 out of 15 times over in rainland. However, on the other side, where I'm going to predict that it doesn't rain, and by the way, how I pick that is how many dots are there, right? All the dots over here are red. More of the dots are blue on this side, at least if you use the counts. So this one's wrong more of the time. And what we look at overall, or at least almost what we look at overall, and I'll get to that almost in a minute, is the misclassification rate. How many did we get wrong? Right? So you, of course, could do how many do I get right instead, but we like to talk about when the model is wrong for some reason. So we use this. All right, well, maybe if I move the line, it'd be a little better. So move it down. Here at 75, I'm now 36.36% wrong. I'm over here and try the model where it does well on not raining predictions. I'm now 19% wrong. And we pick the optimal one. So the algorithm goes through and looks and scans and finds the optimal one. And so that's 17% wrong. Note that because chance of rain is a number in the teens, I actually have a range of possible values here. I picked the average. So then we get a tree. We do this again, right? For each node, we pick which variable to split, which I still haven't quite told you about yet. And then we pick this split by this method. And note that variables can show up more than once. There are actually multiple algorithms for this. This is the correlation and regression tree one. You have a lot of choices about how things work. But this is a model that works like before. So if I have 50% chance of rain and it's cloudy, I'd go through the same algorithm, right? Is the chance of rain less than 55? Yeah, it is. Is the chance of rain less than 15? No, it's not. Is it cloudy? Or is it not cloudy? No, it is not. It's predicting rain.
It's not cloudy.
Yeah, it's not cloudy. Cloudy equals not cloudy is how it's writing it, this particular thing. How to get your thing to give pretty trees is a good question.
How will this correlation of variables affect
Correlation and classification trees generally are greedy algorithms, so it's going to pick the best one, and it's going to get really stuck on whichever one it thinks is best. Correlated variables, it's going to pretty much include one of them probably, and not the other, which is a problem. That's why we're not done yet, indeed. I can't remember. I don't think having correlated variables in the model is the same problem as you would have with regression, where that's really bad for the modeling. All right. So let's talk a little bit about how we pick the variable, right? I've told you how we make a split. How we look for variables is we actually look for which variables have the best split, right? Which would most reduce the error if we used it. So at each step, we look at our node and we say, okay, here are all the variables, here are all the splits. Which one would make the error go down the most? That's the one I'm going to use. And I should mention why I keep saying the word almost, or I'm slightly lying to you. We'd love to use the misclassification rate for error. We can't. That's not the error that's actually used. The reason is misclassification looks like an upside down absolute value. It's pointy. The top point is a place where the derivative doesn't exist. This is not good for computer algorithms. Computer algorithms really like things that tend to work the best in conditions where everything is nice and smooth. So we actually, the two error methods that are common here, Gini and the one called entropy, cross entropy information, that one has a lot of names. Both of those things are approximates of that curve, right? They are formulas that are fancy looking that really just make a shape that makes a continuous and differentiable approximation of this. And they mostly yield the same tree. It's been proven that only in a very small subcase do they make different choices of what tree to make. All right, and then getting back to the correlated variables business, why is this talk not classification trees and predicting fracking? Why are we going on and talking about random forests? And the answer is using a single tree doesn't work very well. Here I have a subset of my data, and I have a nice tree that was generated. It exactly predicts all of the data I've given it. It's probably really bad at predicting anything else, right? It's overfit. Single classification trees tend to overfit rather badly. It's kind of like if I had a scatter plot of a bunch of lines, and instead of drawing a best fit line, I drew a line, that curve that went through all the dots. It does great on the data I've got. It's going to do horrible on future data. That's true here too, for different reasons, but similar problem. So one tree is not enough. So we're going to make a random forest. First of all, mathematically, a forest is a collection of tree graphs. Yes, mathematicians, we make jokes about trees and our tree stuff. And the random part comes from how the trees are made. And I'll tell you about that. All right, historically, by the way, trees have been around since the Neoplatonic philosopher and logician Porphyry in 270 BC. He actually used it to classify things in a weird sort of semi-kingdom phylum sort of object. Here's the classification of Plato, where he thought about things in different categories and used it to classify animals and organisms and people. The statistical decision trees, that method that we're looking at, the one we just looked at, is from 1963 by Morgan and Santos. The random forest was discovered independently. The idea was by Ho in 1995 and independently by Emmett and Gaiman in 1997. And then the algorithm, the one that we're actually talking about, which is implemented in the R-part package in R, is by Brayman in 99. And as you may have guessed from the description of how actually classification trees work, classification trees are pretty computationally intensive. Random forest, where we generate a bunch of classification trees, is really computationally intensive. Although computers have cut up, when I first started using these, it would take a couple hours to run something on a good sized data set. Now it's a few minutes. It's not bad. All right, so how do we make lots of trees? Well, to start, what we're going to do is restrict access to the variables. We're going to say, hey, I'm going to take a random sample of my variables. I'll make a tree from that today. Right, for my predictors. I keep the response the same, of course. And then I'm going to do that again and again and again. I'm going to take a random sample of my variables. I'll make a tree. I'll take a random sample of variables and make a tree. This will allow us to use all the predictors that are good at some point. Because random forests, classification tree algorithms tend to be greedy, and so they tend to pick the best thing, which means they're going to sometimes miss out on the second best thing entirely. And this will allow when the best thing is not in the picture because we haven't selected it to go. And then we usually combine this with some sort of method of validation. The simplest method of validation is to split your data into training and testing sets before you go, before you make it, and make your model on the training test and then test it on the testing test. Yeah?
When you select a random subset data to control, how do you decide on the optimal size of the subset?
It's usually, so at some level, there are preferences, but there are not strong, fast rules. It's usually about square root of the number of variables. Why that, I'm not absolutely sure, actually. I have not read something that says this is why this number, but yeah, that is the general one. And you usually make a few thousand trees. And note that there are other ways to do validation testing here, or you can use cross-validation here. The idea of a testing and training set, or a testing, training, and validation set, if you're trying lots of models, you don't want to overfit your testing set either, is a common thing to any sort of modeling problem, not just random forests. And you can do prediction here. Here's a bunch of trees from the shall I bring my umbrella problem that I'll admit that I made up. Right, but if we have 30 and yes, as in it's raining, it's cloudy and it's 30% chance of rain, I can go through the trees and predict, right, it's less than 40 and it's cloudy, less than 50, it's cloudy and it's less than 55, and get a prediction from each tree. And then you have actually a choice here, but the simplest way to pick what did we just get is to say the majority said no, therefore there's no rain predicted. Note there are other algorithms for doing the voting here. It's different if these are numbers, although that is a possible thing to use as well. But so once you have a random forest, you can use it to predict just like you can a single tree. All right, so let's come back to fracking. All right, we're going to look at our response. Our response now is going to be active fracking or not. So now we're on to our actual statistical problem. Our predictors, we're going to stick in the counts of bacteria. And we're going to ask which bacteria are good predictors of active fracking. All right, so we get trees like this one, the one we saw at the beginning, for each thing. And then we're going to look over the random forest, which variables appear in the trees that we got. Of those variables, how much did they reduce the error each time? What on average did they reduce the error? And then we're going to rank the variables by how much they reduce the error on average. Right, and of course they reduce the error a lot. They're going to appear in a lot of trees. So here we have the list of the top 10, I believe, of the bacteria in the predictive model of whether there's active fracking or not. So note that thing labeled Gini average is the average error. You can think of it as the error decrease if that makes it easier for you. Higher is better. And you can see that the top ones are Sphygobacteriales, Certificaceae, and Coelobacter. And you can, of course, continue this list on down. Where to cut off the list is one of those no hard and fast rule sort of thing. You look for breaks and elbows and certain numbers of numbers to fast rule.
So can I ask a question?
Yeah, sure.
So does that mean the Sphygobacteriales are good predictors or among the best predictors?
Among the best predictors, because we're averaging how well they reduce error, but if they reduce error well, they're likely to appear in the trees too, because that's how we pick which ones go, which ones are node splits. So yeah, these are the ones that are the best predictors. All right, so let's return to the biology. Outside of the bacteria, pH and the other parts of the analysis, because this is one analysis of a fairly large scope of a paper, pH was the most influential variable, followed by other things about predicting the drilling site. If you included in the random forest something like number of well pads, that was a good predictor as well, which is not surprising. Sphygobacteriales, Cytophageaceae, and Coelobacteriales were the most predictive bacteria. And since those are acid lover bacteria and the sites where fracking is active have a lower pH, it's a question of whether the outwash is actually affecting the, like if it's just that the pH lower is affecting the community, or is there something else in the outwash that's also affecting?
So with the Sphygobacteriales,
Yeah.
They are known for being able to degrade xenobiotics.
Mm-hmm.
And that's one of their fascinating characteristics. They're often found in toxic soil sites, that kind of thing, in really high numbers. And they're known for degrading compounds that other bacteria.
Right.
That makes sense too.
And there's some stuff about whether or not the stuff that the outwash from a well includes some of those biocides that are in there, and that some of the predictive bacteria in some of the models, we have new models we have here, that they the degradation of things that we think are actually in the undisclosed chemicals used in fracking list are degraded by some of these things. So, yeah. All right. So this is an ongoing project, actually. So this is about a paper that we published a bit ago. Juniata is leading a new NSF grant from CBET, the Environmental Engineering, with some collaborators from Michigan Technical and UT Knoxville. They just collected new samples this summer. They did an expanded range of samples because you notice that the number of wells in PA, there's a lot of wells in PA. And this time, instead of just taking data from the same place in the site, they now intentionally are doing upstream of the fracking and downstream of the fracking. So we have sort of almost matched pairs-ish sort of object. I mean, they spent a lot of time trying to make sure that they could control for the type of watershed in the first sampling process so that that wasn't the major effect. But having upstream and downstream things of fracking will give us extra data. And of course, you want to know, are the bacteria doing what they do, right? So we're going to look at the function of the bacteria that are present with metatranscriptomics. And we're also taking additional chemical measurements of water. Fracking's been associated with methane, for example, so looking at dissolved methane in the water. So this is actually an ongoing project. The analysis of this data is currently ongoing. I have one slide of that. And I think the functional stuff just came back from the lab recently. So this is actually the current one. So keep in mind that this is family level, I think. It'll tell me. And that they don't look the same as before. And of course, this is family level. There's other things here. Subgroup 6 is a type of phylum of Acinetobacteria, which is acid tolerant. A couple of the alpha protobacteria, the Colobacteriales and the Xanthobacteriales, nope, that's not what I wanted to do, are supposed degraders of one of the chemicals that they think is actually involved with fracking. This is, of course, under the new way where we're using ASV instead of OTUs, and we're using a different library than green genes. And now I don't remember the name of the new one. That's not my end of the pipeline. But so we're looking at this, and so this is an output of a different random forest, where on the bottom we got the mean decrease in error again. Gini is the more commonly used one. As far as I could tell, that's because it runs faster, because information involves a log in its calculation of error. And you can see that first one is the Acinetobacteria is rather big. And the ones that appear multiple times are in different further down the classification. So this is what we're currently working on.
I have a question indeed, so it's a technical question. What kind of package do you use? Because we have too many packages and everyone deal with random forest in different way. So some package, you have to split your data into 30-70 and run on random forest in 70% and this is 31.
Yeah.
However, we are interested on the most important taxa here. So I would prefer to use all of my data. And
Right.
That's why I get used just random forest. And instead of splitting my data, I used out of bag error.
Right.
And,
Yeah.
and that which is
A cross-validation technique.
a cross validation, which is really fast in computation rather than other package like and all of things. So what advantage of using out of bag error versus splitting my data into 70-30?
So I don't remember which one is. So I'm thinking about. So you have options. You have options in. You can do the 70-30 split. The advantage of that is it runs a little faster on the 70, right? And then you have the 30 to test. It's easier to explain what you did, right? And what you've got. You can use all sorts of fold cross validations, and I think out of bag error is one of them, if I remember correctly.
That's correct.
They have to run more things, right? Because you want to have chances for most things to have been out. It does allow you to use all your data, which is great,
Yes.
right? But you don't have a reserve testing set to validate that it's overfit a little bit more. But the cross-validation process takes care of that.
Yeah.
So I think it's We've chosen to do the testing training split and run it that way because it's faster. That may not matter as much anymore. Certainly, Chime has a new implementation of random forest that involves cross-validation. I haven't run it yet. Somebody in the lab has, but not me. So I think that's becoming more popular. Yeah.
Do you analyze your sediment data and water data all together?
Depends on what we're doing because we have more chemical measurements for water. I am trying to remember. So my student did this analysis. I taught her how to do random forests and we did it. So I did not look at the data quite as much as she did. I believe we did all the data for ours because we weren't including the chemical measurements because the main difference of the water ones is we have slightly more chemical stuff.
So when you do the analysis, do you use the relative abundance information or you just use presence absence?
We use relative abundance. Generally, I think we were actually using raw counts this time.
I'm asking because we're doing something similar and we're looking at the sediment of the water sample and the water fraction, and the community was completely different. So I wonder how merging everything together would affect your outcome because you're essentially looking at different samples.
Right. I do not know that in this case, and I think we were doing it on all, but I'm actually not absolutely sure because this is me talking about a student's analysis. And one of the advantages of this technique is at least can get into the common features of it. You aren't trying to mash them together and then doing a POA plot or something.
You had some really great, like, accessible explanations of that. I would appreciate a lot of that. One thing I wonder, so I know for like a lot of multivariate methods, they say it's more difficult to work with microbiome data because there's just too many features. In this, I'm not sure if I understand, like, is it better to have more features, more taxa, or is it better to have fewer taxa?
Generally for random forests, you actually want a large number of features. Or at least random forests work great on a large number of features. For example, I mean, to be kind of silly about it, right? I'm not going to do a random forest on the rain problem because I got two predictors. Like I'm going to pick one of them and then the other, and then done. It works best if you can have a large number of different random samples. And to do that, you probably want a large number of data. But I don't actually know that for sure. Yeah.
So is that true then I assume that's true in terms of maximizing your error reduction, right?
Mm-hmm.
Is having more features better. But what about when it comes time to now reinterpret this in terms of the biological expectation? Mm-hmm. If you say have a smaller subset from your total data that you have mechanistic reasons to suspect that they're involved in some phenomenon, then is there advantages then to restricting to that subset?
And we did that here actually. So we tried running it with the metadata the first time and it said, hey, look, pH and well pads.
Right.
We threw out the active wells because that's equivalent to the other one. Right. pH and well pads, they're great predictors, and so are the water chemistry, the chemistry ones. And then yeah. And so we tried it without it just to get a better thing of the biologicals. So you can do that, yeah.
So you can do that, but what does it do to your predictive value? So here's my thing is that if you did this in Pennsylvania, say,
Mm-hmm.
and you use all the features, and then you come up with a fairly good predictive model for it in Pennsylvania, would it improve your prediction, your generalizability of your predictive ability if you stuck to those smaller subset that you say you had knowledge about?
I don't know. Well, first of all, while we do say prediction is a goal, like for fracking in particular, we can literally go see if the well is running.
Right.
So description is a much bigger goal of like, what effect is this actually having? Whereas predictor is a side
the
thing.
generalizability of your description, then, let's say.
Right, the generalizability of your description. So you're asking me if I include less variables, will I get a more generalizable model?
Yeah.
I don't know.
At least less variables that there's other information, outside information that would lead you to believe that they're mechanistically involved, right? Because you could have, okay, so obviously the bacteria are not actually determining whether there's fracking there.
Correct.
But they're directly responding to whether there's fracking
Yes.
there. But you get other ones that are spuriously
course.
correlated with that.
Yes.
Right?
So what I don't know, and the reason I'm having trouble answering your question, we're trying to look at Texas, for example, since that's one of the areas of fracking. I don't know how much now I'm going to use a biology term and worry that I got the wrong, that the beta diversity between the Pennsylvania and Texas is different anyway. Like, I suspect it is. And so the question is, what biologically does that mean that the same acid-loving bacteria are likely to show up? I don't know that answer to that question. So,
So it might be not taxonomic related, but rather functional bacteria.
right, yeah, it might be. That's not an area, I mean, now we're talking biology and we get into the danger zone where I can
No,
say
I'm just
things
very-
but not know them.
They're trying to think
This
of
is the
some
same
sorts
place
of
to do
these
that, by
problems.
Oh, good, good.
Yeah.
Yes, I can say lots of words, and I know what most of them mean, but I mostly study statistics.
So, I think quite a few of us have done random forests before in our data sets. In tutorials that I found always do some sort of normalization step of the OTU table.
Mm-hmm.
Can you explain why you decided to go with raw counts?
Nobody's asked me that one. At the time, I think I'm pretty sure we used raw counts. I should say that. At the time, I think we weren't sure which normalization method to use, and we had read something that it might work better on actual counts rather than, like the CSS scaling, I'm not sure, so I'm trying to remember. This project we graduated a few years ago, so. I do not remember why we chose to do that, but certainly you can do some scaling first.
But you don't have to.
I don't think so.
Random forest is fine with.
Random forest is fine with whatever you feed it, right? It doesn't expect the numbers to be there. I mean, the advantage of scaling is that you deal somewhat with that overcounting problem you get from read depth, right?
Yeah, but if you're already working with relative abundance, then
Mm-hmm.
it wouldn't really matter that much.
Yeah.
Right?
No. I mean, it'll matter some. It'll get rid of some of the differences between numbers, but at some level, you're taking that picture where we're trying to pick the divide and going squish to it more than you are. Because this technique is more about placement than it is about, right, this is not an outlier, outlier heavily influenced technique, if I'm remembering exactly. So it's I don't think it matters as much here. There are places where, of course, it does.
Did you say that those taxa that are most influential are usually highly abundant?
I didn't do that end of analysis. I don't remember. I know we did stuff with that. I mean, there were lots and all that sort of things.
Not necessarily.
Not
Exactly.
necessarily. Not
In that
necessarily.
case, we wouldn't care that much about normalization.
Yeah.
If they are rare, then, if you sequence deep, then you discover them, otherwise you don't. That might affect.
It might. I'm not sure, actually. I'd have to think about that too. Yeah, go for it.
Somewhat related to this one, but did you detect anything in frac sites that were only detected in frac sites?
I do not know that. I don't remember that being in the paper, but since I have never looked at the whole data, I don't know if that's true or not.
What kind of threshold do you accept the classification or not? Is it 27, 70%?
You mean when do we think that the classification trees are?
Correct.
So there are two things we look at with classification trees. So for example, if I can, it's further down in my notes. So this model was 83% correct on the test data. Right, and so we can compare that to, is it better than random chance? That's kind of a low bar. Is it better than 50% correct? Great. What we most often like to compare it to is it better than the model of just predicting one thing all the time? Right, if my data is 60% fracked, and I do not know for this data what the percentage of fracked is, does it get it right better than 60%? Because I would get it 60% right by just declaring them all to be fracked. Right, so when we're looking at that, and then there are some averaging methods between those two of where the standard is my model good enough to accept it. But there's no like p-value sort of object for these things,
Yeah,
as far as I know.
area under cover, I know it's quite used a lot in that.
Right, AOC is, yeah,
But still, we don't have a threshold to say this is acceptable or not acceptable.
Correct.
It's a gray area. And,
And we
it's really
don't-
subjective based on the person.
and we don't have a great acceptable of like what mean, Grace, and Gini should we look at? Like,
Yeah.
do we say, okay, it's only that first one, or do I take the break here? There's no nice numbers about that either. So. Yeah.
These are mean decrease in Gini index.
Mhm.
Do you have any criteria for the absolute value of the Gini index that would be desirable to have for your, most
So,
inflammatory?
if
types
I'm,
of-
so
So,
let me
okay,
think.
Just let me, so.
Yeah, go for it.
I noticed that you had a list of OTUs, and then the top Gini index for the Spiegel bacteria that was the most influential was 0.14, and then the next one was 0.11. So
Yeah.
do you have certain criteria? I mean, if they all have super low Gini indexes, is it even worth it to pursue this?
That's a good question. I've never seen that anywhere, so I don't know.
Okay.
We have this problem.
Yeah, you have this problem.
I think we should make one up now.
Yeah, so this is one of the disadvantages of classification and regression trees is that there aren't like, as clear as, is the model valid is not as clear as it would before. And I don't know, I don't know anything about that. Yeah,
Could you do like a Berlin alpha or alpha with the two patents?
I don't know.
And that is-
I don't. I have not seen anything like that. I've gone digging around, and we've spent some time trying to find, more directive methods for picking where to cut off, and we have not found anything nice yet. So I'm guessing that either I'm not looking in the right place or it doesn't exist yet.
So this is interesting, I was just sidelining from Terry, is that we probably have enough data in the Microbiome Center to actually ask. Among us, with all our different data sets, that would say, this is what I think, this is for our data that we see, and then pulling those data.
Right, and the one thing I was trying to think of, but
Is that a- something?
I'm trying to remember if the scale of the data affects the error here. I don't think so. I think it's all proportioned out well, but I'm not sure about that part without looking it up. That one I can figure out. But that matters, of course. Right, if it matters on the scale of your data what these numbers come out to, then the scale has to be taken into account first.
I think it depends also on whether you accept the model or not. If the model fails to classify your samples, Gini here number doesn't make any sense because these predictors, however they have higher Gini index, they fail to classify your data.
Right.
So it's meaningless here.
You usually look at what is called the confusion matrix first, like how much correct classifications do you get or not on your test data, right? Or the equivalent of that in cross-validation land. Yeah, and so in order to look at validity, before you're looking at these, you're looking, did my model actually classify better than either I'm going to guess the same thing all the time model, the null model, or random chance at least. So at least there is some check before you go here. But yeah, I don't know what the scale of those numbers. I mean, it's very close to the misclassification rate. So, right, so these should be decimals of some kind. I think, I'm trying to remember where the scaling is, the formula actually right now.
Could you go back to the other thing?
Yeah. Which of course is at the end.
So what I've done to validate this thing, I did indicator species analysis, which figure out which kind of taxa indicating this condition and this condition, and also did differential taxa abundance and made a Venn diagram to grab the most bacteria that all tests have been agreed that these bacteria are influencing my variables, and then work on these core bacteria outside.
Right, and this was part of our stuff was part of a larger analysis. So the random forest was one bit of a larger paper that included co-occurrence networks, some differential abundances stuff, some left things. There were other things there that I wasn't. Yeah.
So, okay. So an alternative way of doing the classification, or what are important predictors of classification is LEFSA, for instance.
Mm-hmm.
Do you tend to get the same top hits?
So my memory right now is that LEFSA, its most differential is somewhere down in the list. It's not towards, well, not in this list, the previous one, because this is the start of the analysis of the new data. So the other one. In this one. The top thing in LEFSA was somewhere in this list in the top 20, and I don't remember where it is or which one it is right now without looking at my notes. But yes, they do at least show up. Are they the same? No.
And then, so that's been my experience from what I've seen too.
Yeah.
Do you think there's a way to combine those to have sort of?
I don't know. That would be an interesting sort of technique. I'm not sure how much they get at the same signal, and I'd have to think about that a lot. Yeah, that's interesting. I don't know.
And then my other question is, we have to kind of hinted at this before about using presence absence. Is it possible to do random forest using presence absence?
You can use categorical predictors in random forest just like you can use numerical ones. The split becomes instead, of your category options, which ones go on which side and which ones go on the other. So a categorical like presence absence would be a very different model because you would have no scale and of counting, but it might be worth trying. I don't know if that's better or worse.
Yeah, well, there's just so
I haven't seen that.
So for instance, for yours, I don't think it would, for this fracking story, I don't think it would make sense to go that way. But there's other things. So like one of the problems we're looking at is can you predict that someone will have an increase in this metabolite if they consume this food? Okay. And so with human gut microbiomes, people have different microbiomes. Some have this species that there's a lot of functional overlap, but then there's a lot of taxonomic divergence. So does it matter which bacteria you have performing this function? And so there's a lot of, it's just totally not there in this person, but they might have some other functional equivalent. Can we go through then and say, okay, if you have this food degrading bacteria and you have this metabolite producing bacteria, you will get that metabolite when you eat that food, versus if that's not present, then you don't.
Right.
I think we can try and answer that type of question.
Right. That seems like a more like that you draw a line of what is a presence affected by the thing rather than the baseline.
Yeah,
and
because
so that might be more useful.
So the thing is, if
Background
you go by relative abundances or just counts or whatever, the problem is that if you keep eating that food, those counts go up. But it could have started at a very low level, but you can now essentially enrich for that bacteria over time by putting in that food. Whereas if it's not there at all, of course you can't enrich for something that's not there. That's why we're kind of play around with that idea there. But I didn't know how amenable the force would be to answering that question.
It certainly can use categorical predictors just like the others. You lose a little bit of the flexibility of where the line is drawn. So I imagine that would make it overfit more, but random forest is supposed to compensate for that. So
that would be your concern is overfitting.
Probably, because right when you think about it, you're not drawing a line between things and then picking an error based on that. You essentially don't have a choice, right? If you've got two conditions, one's on one, one's on the other.
Yeah,
and you don't just split.
Whether or not
run
to
faster.
use it in the tree, right?
Yeah, you can choose whether or not to roost it on the tree, and it would do that based on the error.
Yeah.
So I don't see why not. I don't think I've seen that. On the other hand,
but then in that case, you probably predict that it's important to have a larger number of features then.
yeah, probably.
So, just, I don't know if you know this, but patch loss is a lot of random forest with
I know.
color.
Yeah.
Where
they are usually using relative abundance data in there, although what they saw, some of the strongest predictors were with if you didn't have certain bacteria. But I think when they were actually constructing their random forest, that they were actually using the abundance rather than actually breaking it down to presence absence.
Oh, yeah.
But what they did, and you're right, is that what they did end up finding was certain bacteria not being there were the strongest predictors of
Well, thank you very much for stimulating that discussion and for presenting your work.